All right, everybody, here we are, and we're talking about meiosis, or the process of gamete formation, to which cells produce sexual reproductive cells, which are called haploid cells, a special type of cells. And as you can see, this involves two different division processes, which will end up reducing the total number of chromosomes in half. And that's what the meiosis is all about. But before we can actually start talking about meiosis, we need to do an introduction to the different kinds of cells and chromosome numbers involved in the meiotic process. So let's talk about that. So for starters, when you think about your body and the cells in your body, you think about somatic cells. Somatic cells is another word for all the cells in your body involved in normal cellular activities, such as, for example, your nerve cells, your fat cells, your muscle cells, bone cells, digestive cells, epithelial cells, all the tissues of your body which are involved in normal anatomical and physiological processes. Now, when these cells divide, they divide by mitosis, always. That's all they can do. And they do so in order to grow, develop, and also to repair themselves. And so mitosis in multicellular organisms is mostly for growth and development. Remember, though, that some primitive eukaryotes, such as protozoa and algae and mold, also do mitosis for asexual reproduction. But anyways, when it comes to sexual reproduction in animals, you're going to need something called germ cells. Now, germ cells are a special group of cells which, unlike somatic cells, can actually undergo a special process called meiosis. Only germ cells can undergo these processes, and they can only be found in the actual sexual organs of the males and females. So the germ cells will be in the male testes and in the female ovaries. You can see the male testes on the top and the male ovaries on the bottom here in a histological picture that shows you the tissue. And you see in the middle there the actual maturation of cellular cells which are undergoing meiosis, and which is what we're going to be talking about today. So remember, somatic cells are the only cells which can germ cells are the only cells that undergo meiosis, and they are going to be in the sexual organs of males and females. And then somatic cells all reproduce by mitosis. Now remember that before any germ cell undergoes meiosis, it must first undergo mitosis, because if you get a germ cell and you do meiosis, you're gonna lose that germ cell, and it will no longer be able to undergo future meiosis processes because it gets split up into four. And so, in order to preserve the total number of germ cells, you always double one, and then you take one of those and undergo meiosis with that one, and you have the other one left to repeat the process again. So that's kind of how it works with germ cells. So again, you would have one germ cell which will split up by mitosis into two germ cells, like we talked about in the previous video, so a regular cell cycle. Now this one will stay so that the process can repeat itself, but this one will actually undergo meiosis to form the gametes, right? And then if I want to repeat the process, I can keep going and going, but making sure that I always create one more germ cell so that I always have more germ cells to repeat the process. All right, and that's kind of how it works in your body. Now, when germ cells undergo meiosis, they actually make gametes. Now, gametes are special kinds of cells which are called haploid cells. And gametes are involved in the actual sexual reproduction. You see here in the top right corner the fertilization action of thousands and thousands of sperm trying to win the battle to be the first one to get to the egg. In the bottom, you see representations of eggs and sperm and how they look like. And then you see the diagrams of the actual parts of the egg and the sperm. And I like to talk about that, and to see that the sperm has a long flagella-like tail which propels the sperm towards the egg inside the woman. And you would notice that that tail will be rich in mitochondria to produce the energy necessary to flap that tail. And then you have this protective head, which includes an acrosome, which is an area full of digestive enzymes, which are in charge of perforating the outside of the egg, including the jelly coat, which protects it. Inside the egg, you're going to have a polar body, which is a remnant of a cell that gave up its cytoplasm to enhance the cytoplasm of the egg. We'll be talking about that when we do oogenesis, or egg formation. And then you have the nucleus of the egg, which stores the genetic information coming from mom. And inside the cytoplasm of the egg, you have these little yolk droplets, which are full of protein stored to power up the initial cell divisions of the embryonic process. Now remember, the nucleus of the ovum and the nucleus of the sperm are only half of the nucleus of each respective parents. It's by when they're joining that you form a nucleus of, there's going to go ahead and form a zygote that divides to form who we are. So a different way of looking at that, we're going to see a picture of that very shortly. But anyways, before we keep going, now we also have to talk about chromosomes number. But before we talk about chromosome number, let's talk about chromosomes. Now remember, the chromosomes are these linear DNA molecules which show up in your karyotic cells, and there's one continuous DNA molecule coiled up in a series of around proteins called histones into coils and supercoils and things like that. So, and then each of these continuous chromosome molecules includes thousands of genes, and you can see here represented on the top right corner, each band is supposedly representing one gene or one gene sequence. So you see that there could be thousands of genes on each of these chromosomes. Now, a typical human will have 46 total chromosomes, but notice that they are paired, and so you actually have 23 types of chromosomes, but 46 total. So even though you have 46, you only have 23 types. So I think of this as a library of genetic information, or an encyclopedia of genetic information. Back in the day, before the internet, when you needed to find out reference, you would go to the library and open these encyclopedias with volumes of information. And then inside each volume, you would find certain kinds of information. And inside each of those volumes, there you have chapters devoted to certain pieces of information. So think of it that way. We have an encyclopedia of genetic information. And within those encyclopedias, within those portals, there are smaller websites which store the information about a certain topics, which are the genes inside of each chromosome. But each chromosome in itself is a little encyclopedia, or a little book out of an encyclopedia of genetic information. But you get a book from mom and a book from dad about the same topic. So, for example, when it comes to knowing how to make an eye, part of the information that makes the eye comes from mom, and part of the information comes from dad. Both have a say on how the eye is made. So you have the dad's book on how to make the eye and the mom's book on how to make the eye, and then the way you make the eye is about a relationship between those two things, which we're going to talk about when we do Mendelian genetics later in the year. Now, what's important about this is this idea of homologous pair, or homologs. Chromosomes are paired in the human karyotype. Remember, karyotype is a picture of the human genome, or all the chromosomes inside of a cell lined up by size. So from 1 to 23 in numerical order by size. So 1 being the largest and 23 being the smallest. The first 22 sets, as you can see here in the bottom, are called autosomes, or these are the chromosomes which are involved in all the bodily functions of your body. And then the last pair is called the sex chromosomes. Now, if you're like this karyotype here, and you have an X and a Y, you're male, because the Y chromosome causes the maleness to occur. But if you have two X's instead of having one of that, you would have two copies of the X, you would be female. So both of these karyotypes represent male karyotypes. So a female karyotype will have a second X instead of a Y. All right. Now notice that there's a slight difference between these two karyotypes. This karyotype up here includes a singular chromatid, or a single chromosome structure, and then you have pairs, just like it looks in this picture here. Now, these chromosomes will never actually look like that because they don't really coil up until after the S phase, right before mitosis. And by then, the chromosome has replicated. So between the top and the bottom here, what's happening is the synthesis phase of the interphase cycle, of the cell cycle, where each chromosome or continuous DNA molecule doubles itself up. So, for example, what they did here, in the top right corner, you have two examples of homologs. The maternal 11 and the paternal 11, the maternal 17, the paternal 17. They're just showing you how each chromosome type is paired, which you can also see in the karyotypes that you can see here in the left side. By the way, karyotype means the types of DNA, or the type of nucleus, or a picture of the nucleus, ordered by size. So what will happen is that what they did here in this drawing is that they picked up that chromosome and they blew it up, and they saw that the maternal one duplicated to make 2, and the paternal one duplicated to make 2. And so what you end up with, you still have the homologs. So these are homologs. They're books about the same thing, But now instead of having 2 chromatids, you have 4 chromatids, but these 2 chromatids here are sisters in the sense that they're exactly the same thing. Both are copies of the maternal. These 2 are both copies of the paternal. All right? I know the color is green, but it has nothing to do with the chromosome 17 you see on the top there. All right? So they're just showing you that during the S phase, the chromatids will double up and become sister chromatids. So when you actually see an actual karyotype, when the actual chromosomes, they will look a little like this. So you have paired up axes, not paired up lines like you see on the karyotype on the top. This karyotype here will only be possible if you induce the chromatin to call itself up before mitosis or before the S phase takes place. So a karyotype like this will only be available during the G1 phase before the DNA starts to copy itself up. But as soon as the synthesis phase starts, the karyotype starts looking like that. Also notice that they order the karyotype by in groups of each sets of chromosomes are formed letter groups, all right, from A to G. And that's only important when we do genetic analysis later in the year, all right? Now, this is basically chromosome families, all right? Now, anyways, this more important part of this is to understand the idea of homolog pairs, in which chromosomes are paired up. And this is what our typical eukaryotic karyotype will look like. The human karyotype is the one that we've been talking about on this slide. Now, cells in eukaryotes or animals will undergo a cyclic process where the adults, by meiosis, which is the topic that we're talking about now, will create the actual gametes. So the germ cells of adult animals which underwent puberty will, or mature adults, will produce the egg and the sperm. The females make the egg or ova, and the males make sperm or spermatozoids. And then these things will then fertilize or join to again form a zygote or an embryo, which is going to duplicate to go back to an adult when the cycle repeats itself. Now let's look at the chromosomes numbers here, all right? The adult, like we said before, is a 2N or 46 chromosomes. What do we call about that? Why 2N? Because you have 2 copies of each chromosome type. So in humans, you have 2 copies of each chromosome type. There's 23 types, so you have a total of 46. So the chromosome number for humans is 2N equals 46, right? Now, when meiosis happens, and we're going to be talking about this in the next video, you actually reduce that number in half. So the sperm is not a copy of that, but only half of that. And the egg is not a copy of mom, but only half of mom. And so these cells are just N cells, or half cells. And they're, instead of having 46 chromosomes, they will have 23. Basically, they only have 1 cell of each type. So they have 1 cell of each type. And since we have 23 types, it will be N equals 23 for these haploid cells. So there you have it. A normal adult animal is diploid and has 2 copies of each chromosome, like we saw in the previous slide. But the gamete cells will undergo meiosis, which reduces the chromosomes numbers to a haploid. Now, once fertilization happens, the addition of the egg and the sperm reestablishes by fertilization a 2N cell called zygote. And then that 2N cell will undergo mitosis through the process we explained in the previous videos and become again an adult that has multiple multicellular diploid cells. So that is basically how the process works. Now, remember that only germ cells can undergo meiosis, and normal somatic cells are the ones which undergo mitosis. Now, lastly, and this is going to be important when we talk about plant evolution, some organisms are capable of, instead of having 2 copies of each chromosome, the way it looked on the karyotype we saw on the previous slide, they will have 3 copies of each chromosome, like you see here, or 4 copies of each chromosome, like you see there. So these are called triploid, or tetraploid, or pentaploid, or ectoploid, or octoploid. You can have as many copies as possible. Now, in plants, this is going to be an important thing. And that is called polyploidy. When you have multiple copies of the entire set of chromosomes, is, instead of being 2, is 3 or 4 copies for each type of chromosome. So in that case, instead of being 2N cells, there'll be 3N or 4N cells. Another variation is when you call aneuploidy. And that happens because of problems during meiosis, where instead of having pairs like this, you would get something like a chromosome missing, like that. So see, chromosome 1 is missing. or maybe an extra chromosome like that. So this is called trisomy or monosomy, and we'll talk more about that when we finish meiosis. All right?